Well, good afternoon, everybody. Thank you for being here, joining us to this first session. I am Piga Fernandez. I'm a GIST patient. I'm the Global Relations Coordinator for the LifeRev Group. And I have been honored to introduce you to David Josephy. David is a retired professor from the Department of Molecular and Cellular Biology of the University of Guelph, Ontario, where he taught since 1983. His research interests include molecular toxicology, drug metabolism, and cancer. He has been active in the LifeRev Group since its inception. Josephy is a member of the LifeRev Group science team and president of the GIST, Sarcoma, and LifeRev Group Canada. Between 2011 and 2012, he spent a sabbatical leave working in the GIST research lab of Dr. Jonathan Fletcher at the Harvard Medical School. David is a great friend of the LifeRev Group, an extraordinary photographer, and an amazing bird watcher. So, David, the microphone is yours.
Thank you, Pia. And where do I go now?
I'll do that for you.
So this is a presentation I've given quite a few times now, but it evolves a little bit every time, okay? So as the science and medicine evolves and as my thinking evolves a bit. So let me say, first of all, I'm a biochemist, but I'm not a medical doctor, so I can't give you medical advice of any kind. And the other disclaimer I have here, which is an important one, is that in biology and medicine, there are rules, but there are always exceptions to every rule. And so many of the statements here are simplified or made more cut and dried than they really are. So please bear that in mind. Okay, so I'm going to begin with the little top 10 list that we put together a few years ago. This is like if you missed the rest of the talk. But these are a few, I think, important points that you want to bear in mind. So first of all, the GIST, and I'll do this very quickly, and then we will elaborate on some of this as we go along, all right? So GIST strikes randomly. Getting GIST was not your fault. Okay, I'll expand on that later. Unlike most common cancers and most common GI cancers, GISTs are now very treatable. Very treatable, more treatable every year. GISTs are still rare. They're not ultra rare in medical jargon, but they're rare. And so we advise everyone with GIST to find a GIST specialist. As we always say, if you own a Lamborghini, well, you shouldn't, but if you do own a Lamborghini, you don't take it to the Ford dealer for maintenance, okay? Very important to know whether your GIST is localized or metastatic. That's a fundamental branch point in GIST treatment. It makes a huge difference to everything else. Get a copy of your pathology report. It's your report. You have a right to see it, get it. Normally, there's no trouble getting it, but make sure you do get it. Most GIST patients now, at least in North America, are getting mutational testing done, but not all. Ask to get mutational testing done. Ask your doctor. Don't take no for an answer. For localized GIST, most patients are getting, not all, but most patients get adjuvant imatinib, essentially prophylactic imatinib to stave off regression. So that's progression and recurrence. So you really want to think about that. There are now 4 on-label take-home drugs for GIST. There are also a lot of other off-label treatments, as we say, but there are 4 different drugs. We'll talk about them later. All drugs have side effects, including all the drugs for GIST, but most of these side effects can be managed, in most cases, pretty well. And number 10, reach out and join a support group like this one. It's surprising still that only a small minority of patients do that. And we know that patients who join a support group have a better prognosis than ones who don't. That doesn't actually prove that joining a support group will improve your prognosis. It's correlation, not necessarily causation, but it doesn't hurt. So please join the group. Okay, I want to talk, and every topic will be covered briefly, but let's talk about what causes GIST. That takes 10 seconds because we don't know. What are ICCs? I'll explain that terminology. That's where GISTs come from as in a cell biological analysis. We'll talk about KIT, explain what it is. I want to talk, this is important, about what mutational testing is and how it differs from IHC, which I will explain. Talk a bit about the existing drugs, so-called TKI drugs. I'll explain that too. And then we'll talk a little bit about the prospects for new drugs. Things have changed unrecognizably over the past 20 and some years. This is a slide just illustrating, I won't go through it in detail, but just illustrating the rapid development of progress of understanding GIST. So let's recognize first that GIST really was not even recognized as a disease until about 1998. It really wasn't even recognized as a distinctive disease. Any information from before 2000 is completely irrelevant, I would say. Even the later information is rapidly being overtaken by new knowledge and advances. Please remember, every patient is going to look up, hi Dina. Every patient is going to begin by looking up survival statistics. It's understandable. Bear in mind, when you do that, you're looking back in time. You're looking at results today for people who were diagnosed 10 or 15 years ago. It's different today. So always bear that in mind. Okay, and along the bottom of this chart are important scientific advances in understanding GIST. I'm not going to talk about them one by one on this slide, but just bear in mind they're coming along fast, including the endpoint here is 2022. It could make it 2023 or 2024. Even in the last year or two, there have been significant advances in understanding. So the research is still moving fast. All right? That's good. Okay, a very brief note. If you came here to hear about sdh-deficient GIST, you're in the wrong room. sdh-deficient GIST was recognized about 15 years ago as being a distinctive form of GIST. Pardon me. So distinctive that, to me, in my view, it's almost a different disease. There's very little in common between the biology and treatment of GIST and sdh-deficient GIST. So I'm not going to say anything more about that, about sdh-deficient. There is a separate track in this meeting about sdh-deficient GIST, but I'm not going to say anything more about it, okay? From my point of view, it's really a different disease that happens to look similar in some ways, but it's fundamentally different. Okay, what causes GIST? Again, this is a slight oversimplification. Well, let's start with a general statement. GISTs occur sporadically. GIST patients have nothing in common other than having GIST. If you look around the room at any GIST patient meeting, you see that. There's no obvious common feature, ethnically or occupation or geography or whatever. So as far as we know, it strikes entirely at random. And I often say, I've said for years now, if there were any major risk factor, smoking or drinking or where you work or whatever, we would have noticed by now. And we just don't. So GIST is not inherited. It occurs at random. It can't be passed on to your children. There are very rare exceptions to this rule, which are mainly in the category of the sdh-deficient GIST. It's extremely rare cases of inheritable KIT mutant GIST. It's almost unknown. Not quite unknown, but almost unknown. I think 1 or 2 families in the world, really. All right? By the way, if you've got questions, One of the families is here. Yep, yep. Yeah, we met, I think we met at a previous meeting maybe or not? I don't know, yeah. Okay. Inherited KIT mutant GIST. Yeah. Okay. There are exceptions to every rule, and they're sitting in the second row. Okay. Most of you probably are aware of this already, but GIST is a sarcoma, not a carcinoma. 90% of cancers, or so, 90 or so percent, all the common cancers, lung cancer, breast cancer, prostate cancer, colon cancer, skin cancers, most of them, they're all carcinomas. GIST is not. It's an entirely different subcategory of cancers called sarcomas. Sarcomas are carcinomas arise really in lining tissues like the skin, lining of the liver, the lung, the colon, other surfaces, in so-called epithelial tissues. Sarcomas arise really in connective tissues or supportive tissues within the body, not lining tissues. And they're fundamentally very different kind of disease from carcinomas. So when you hear people talking about GIST as a stomach cancer, you should correct them and say, it may occur in the stomach, but it's not a common stomach cancer. Common stomach cancers are carcinomas, and this is not one of them, as we'll talk about in a minute. All right, so GIST is gastrointestinal stromal tumor. It's a sarcoma occurring in the GI tract, and for reasons which will be clear in a minute, it can occur anywhere in principle, anywhere along the GI tract, from the esophagus to the anus. Most commonly in the stomach and the gut, but it can occur anywhere along the GI tract. And there's an explanation for why that is the case. Right? It makes a difference. There's a difference in the biology and medicine and maybe even in the prognosis and treatment, depending on where along the GI tract. Certainly makes a difference to surgeons, makes a huge difference, where along the GI tract the GIST occurs. But I'm not going to get into that in detail. So why does it occur anywhere along the GI tract? It's because the GI tract works by what we call peristalsis. Peristalsis is the muscular contraction waves that move food along the GI tract as you digest. And those waves begin in the esophagus and go all the way down to the end. Those are the waves of contraction every 8 seconds or so, the pulsatile waves that move food along the GI tract. And those waves of muscle contraction are coordinated. And the coordinator, the person who says, stroke, 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 like the coxswain on a boat. The person who coordinates that is a group of neuromuscular cells called ICCs, interstitial cells of Cajal. Cajal is the late great Spanish anatomist who first saw these cells in the nervous system, right? So because the ICCs occur anywhere along the GI tract, that's why GIST can begin anywhere along the GI tract. This, too, is really much more complicated. It turns out there are all kinds of subsets of ICCs and different characteristics, but we're not going to go there. ICCs are where the GI sarcoma begins. Okay. And so just to emphasize again, carcinomas, the common cancers, begin in the lining of an organ, lining of the gut, for example. But the sarcomas begin in the muscular wall outside the lumen of the gut, outside the epithelial tissue. Okay? Don't need to belabor that any further. Okay. So that's a little bit about the biology of where GIST begin. Now let's get into the part I like, the molecular biology. Sorry, that's my thing. So we now understand a lot about the molecular biology of GIST, and that's really important because it's transformed the way that the disease is treated. So first we need to talk about, I think first we should talk about oncogenes. Onco for cancer. So oncogenes are were discovered in the 1970s, really beginning with research at NIH in Washington and leading to Nobel Prizes. Oncogenes are normal human genes which, when they undergo mutations, can give rise to cancers. Oncogenes, every gene, well, not every gene, most genes encode a protein, a functional unit in the cell. And oncogenes encode proteins that act as, a good metaphor is a cellular gas pedal that, or if you like, a master switch that controls the overall behavior of the cell in which the gene is expressed. Okay? And under normal conditions, oncogenes, oncoproteins, the proteins encoded by oncogenes, sit typically in the cell membrane. They listen for a signal coming from the external environment, through the bloodstream, really. And when they hear the appropriate signal, they step on the gas and tell the cell to get activated, including dividing and proliferating. It's, of course, a normal process. The regulated normal cell division is why we're all here. It's how an embryo develops into an adult. It's how we repair wounds. It's how the blood is renewed, red blood cells are renewed. Regulated cell division is normal biology, and that's what oncogenes do. They regulate and control normal biology of cell division. Unfortunately, when a mutation occurs in an oncogene, it can give rise to a mutated or aberrant or variant form of the protein, a protein that no longer listens for the signal. It's lost the ability to listen to the signal. It's what we call a constitutive protein. It's active all the time, whether it should be or not, right? The metaphor is that it's like a gas pedal that's stuck to the floor and is no longer responding to the driver's foot. Okay, and that leads to unregulated cell division, and that can lead to a cancer. This is extremely oversimplified, but it's fundamentally true. It's basically correct. It's a lot more complicated than that, of course, but what I said is basically correct. And we now, in the, almost 50 years of research since the discovery of the first oncogenes, we now know dozens. You could look on Wikipedia and see a list of dozens or even maybe 100 or more different oncogenes, human oncogenes. And they all have alphabet soup names like BRAF, EGFR, HRAS. These are all different human oncogenes. Typically, specific oncogenes are associated with specific kinds of cancers, non-small cell lung cancer or hereditary colon cancer or whatever. It's not an exact correspondence, but it's typically the case that particular oncogenes are related to particular cancers. And so that raised the idea, even 50 years ago, that raised the hypothetical idea, if we can identify the oncogene, and if it really is a stuck gas pedal, maybe we can disconnect that gas pedal. Maybe we can just inactivate it. Maybe with a drug. And for 30 or 40 years, people thought that's a great idea, but it's crazy and it'll never work and you can't do it. Luckily, some people didn't listen to that. Okay. So there are dozens of different human oncogenes, but the one that's of most concern to us is called KIT. It's not the only oncogene associated with GIST by any means, but it's the most important and characteristic, the KIT gene. This was discovered in 1998, and that's why everything before 1998 is irrelevant, because this discovery completely transformed the understanding of GIST. Okay, and to say fundamentally what the relationship is, GIST tumor cells almost always, again, there are exceptions, but almost all of them express a protein called KIT encoded by a gene called KIT oncogene. And fortunately, from the point of view of therapy, very few other cell types in the body do express that protein. That's actually a good thing because that provides some specificity to the treatment. Again, that's oversimplified, but it's not false. In most GISTs, again, not all, but in most GISTs, that KIT oncogene is mutated. It's almost always expressed, usually in mutant form. Not always, but usually, as we'll see. Okay, it took about 25, 30 years of fundamental research to figure this out. In fact, some of that research started near where I live in Toronto with Alan Bernstein, but that's prehistory. But the big breakthrough was made by these two doctors in Japan, to whom we owe everything, actually, who really finally made the connection between the KIT oncogene, which had already been known. I mean, the KIT oncogene was known, GIST was known, but putting those two ideas together, that was these folks. And that made all the difference. Okay, why is it KIT? Sounds like it stands for kitten, and in fact it does. Geneticists have this penchant for giving silly names to genes. They've tried to control that now, but especially the fruit fly geneticists were crazy about this. They have genes with all kinds of crazy names, like son of sevenless and stuff. It's just nutty names. But anyway, geneticists are a bit weird. Anyway, KIT stands for kitten. The oncogene was discovered, as I already alluded to, quite a long time before the connection to GIST was known back in the 1980s. And like a lot of oncogenes, it was first identified as a gene on a little viral genome, on the small genome of a virus that was identified as causing cancer in cats. It was called the feline sarcoma virus. And so the name KIT really refers to, cat was already taken, so they went to KIT. In fact, that's true of a lot of oncogenes. A lot of genes have strange names like that. And a lot of oncogenes were discovered on viruses. So the gene was known and the disease was known, but it wasn't until the Japanese scientists, doctors, figured this out that the connection between that gene and GIST was recognized. All right? Okay, this is Brian Drucker, who works in Oregon, just a little bit north of here. This is a man who deserves a Nobel Prize, and for some weird reason, he still hasn't got it. But one day. So this is the guy who didn't listen to the naysayers and thought, yeah, we really could do it and develop a drug that would target an oncogene. And that oncogene he was interested in, this is a whole different story, is a gene called ABL that is characteristically associated with a particular rare leukemia called CML, chronic myelogenous leukemia, which you don't need to know about. But he realized that this gene, ABL, was associated with CML, and he thought if we can inhibit ABL, maybe we can do something about CML. And that was the research that led to the discovery of the drug called imatinib. And that was one of the great breakthroughs in modern cancer research. It's even celebrated in popular magazines, for example. And it had overnight phenomenal success. And it immediately transformed the treatment and prognosis of that rare leukemia. What on earth has that got to do with GIST? Because the disease is totally unrelated. It's a leukemia. But that's history. So yeah, by 1998, we knew that most GISTs are driven by mutations in KIT. After the KIT oncogene was discovered, then it became clear at the molecular level, at the genetic level, at the DNA sequence level, that the KIT protein is actually quite similar to the ABL oncogene. Not identical. They're like fourth cousins or something like that, right? They're not super close, but they're close enough that you can tell it's the same family, right? So CML is driven by the ABL oncogene, GIST is driven by the KIT oncogene. ABL and KIT are distant cousins. So from 1998, the revolutionary work of Drucker and colleagues, imatinib immediately came into use, widespread use for treating CML. And then a few brave and insightful doctors drew this connection. They said, look, if imatinib works against ABL, what can we lose by trying it against KIT and GIST? They're related after all. There was no reason to be confident that it would work, but there was really nothing to lose, frankly. And so there's a whole interesting story behind how this first trial was carried out, which I don't have time to tell you about, maybe over coffee. But it was tried on one patient, a woman from Finland, all right, who came to Boston and was treated by George Dimitri. And they took the brave decision to try treating that patient with imatinib. Okay? And it was an unbelievable. Luckily, listen, it's possible that that first patient could have been unresponsive to imatinib, and that would have stalled everything for years. But luckily, that patient was, not all GIST patients respond like this, but luckily that patient did. She responded incredibly well to imatinib, unbelievably well. And so these are PET scans from the first publication. The left is before treatment and the right is after. And all you need to know is that all that black stuff going away means the tumor just stopped dividing. The tumor was just stopped in its tracks. It was an amazing result. It was rapidly published in top journal, the New England Journal of Medicine. At the time, I'm told it was the first time in the history of that prestigious journal that a paper had been published as a case report on N equals one patient. And not every patient has responded that well, but it was an amazing result. And that's really where everything began for modern treatment of GIST, right? Okay, so now take a few minutes to talk in a little more detail about KIT and how it's analyzed. So I'll just reiterate then that KIT, and by the way, KIT, like a lot of genes, it goes by some other names because it was discovered by a variety of different routines. So sometimes you'll hear it called c-KIT, which just means cellular KIT. It's the same thing. And pathologists have a whole lot of different terminology for genes. It always begins CD for cluster differentiation. Doesn't matter. But so KIT is also called CD117. So if you look on a pathology report, it's often labeled as CD117, but it's the same thing. It's just different terminology, right? Okay, so the KIT protein is made, as I said before, by only a few types of adult cells, and that includes the ICCs, the pacemaker cells in the gut, and it includes almost every GIST cell. So after a cancer is diagnosed, the next step typically would be surgery, and the surgeon will take a specimen of the tumor or a biopsy or the surgical specimen, a specimen of the tumor, and they send it down to the basement to the pathology lab where a doctor lives in the dark and never sees patients and probably doesn't even know how to talk to a patient, but they're really smart doctors, and all they have is a microscope and a bunch of stains, all right? But they're really pathologists are absolutely crucial in this whole diagnostic process. And so what the pathologist will do is really two things. First of all, they just look at the cells under the microscope. And a good pathologist can just look at a cell and make a pretty good guess as to what it is. It's a fat cell, it's a nerve cell, it's a muscle cell, whatever, just by looking at it under the microscope. They're clever that way. But also, the second thing they do, which is very important, is IHC, immunohistochemistry. Absolutely crucial. So immuno means using an antibody, using the proteins of the immune system. Antibodies recognize specific proteins. Histo means cells, tissues, histology. And chemistry, well, chemistry is chemistry. Right? So the essential step, besides looking at the cells, the pathologist has to do IHC to test which proteins are being expressed by those cells. What is their biology? What proteins are they expressing? And essentially, they will always test whether the cells are expressing KIT. And to do that, they need an antibody that recognizes KIT. And nowadays, you can just buy those antibodies out of a catalog. I mean, it's a whole story as to how you get them, but those antibodies you basically buy from New England Biolabs or any other bunch of biological supply companies. So the pathologist will take a slide, a thin sample of the tumor. They will treat it with this specific antibody that recognizes KIT protein and nothing else. And then they use some biochemical magic to visualize that interaction in a way that turns the sample brown. It's a stain. It's a biological stain. The details you don't need to worry about. And so when they see the tissue sample covered in brown stain, they know that that cell expresses KIT, and that's strong confirmation that it really is a GIST. Okay? This will be done on every suspected GIST sample. Guaranteed. Certain to be done. So when you look at your KIT, at your pathology report, it's going to say that it's KIT positive or CD117 positive in 99% of the cases. It'd be very unusual if it didn't. Right? So in fact, it doesn't tell you anything. I mean, the thing is, it really doesn't tell you anything about treatment or whatever. All it tells you is, yeah, we were right, it really is a GIST. And the doctor was probably pretty sure it was GIST anyway. So it's really just confirmation. It's essential confirmation, but it's really nothing more than confirming, yeah, you're right, it really is a GIST. If it looked like a GIST and was CD117 negative, then they've really got to do some more thinking, but that'd be very rare. Okay, so this really doesn't tell you all that much other than, yeah, it really is, you're right, it really is a GIST. Okay, now we've also mentioned mutational testing. So I put this slide together a few years ago, and I think people have appreciated seeing it because it's a comparison between these two types of tests, which unfortunately tend to get confused in people's minds, but they're very different. Okay? So it's a table that compares, on the left, immunohistochemistry, IHC, which I just talked about, all right? And on the right, mutational testing. So let me just run through this because I think it is important. First of all, what is it? Well, IHC is just staining for the presence of the KIT protein on the cells, like I just said. Mutational testing is DNA testing. It's sequencing of the DNA encoding that gene. So at the molecular level, at the technique level, it's a completely different technique. It's DNA, not protein. So it's just a completely different technique. It just happens to be the same target, KIT. So what does it test for? Immunohistochemistry, IHC, tests for whether the cells under the microscope are expressing KIT protein. Mutational testing tests reads the sequence of that gene, essentially, and tells you whether it is the wild type, normal, quote-unquote, version of the gene, or whether it's mutated. It's a totally different question. Because you can express the protein in the normal protein. Yeah. So what do you learn from that? Well, IHC tells you that the tumor is a GIST. But like I just said, usually we already knew that pretty much. So it's really basically just a confirmation that it really is a GIST. Mutational testing tells you, first of all, whether it's a KIT mutant GIST. And that's only about 3/4 of GIST. There are a lot of GISTs that are not KIT mutant. And that's really important to know. So what do you need? For the IHC, they just need a tumor sample from a surgery or a biopsy. And basically, that's all you need for mutational testing, too. You can even use an old slide that's been kept in the vault, a so-called paraffin-embedded tissue sample. And then the last line is important. Will the test actually be done? Because if they don't do the test, it's not going to do you any good. IHC will always be done, guaranteed. I mean, it'd be medical malpractice if they didn't. Mutational testing is usually done, but not in every case. We really think you should push to get it done, whether or not the doctors are inclined to do it. It's not a big deal nowadays to do the DNA sequencing. Okay? Again, if you want to interrupt with questions, that's fine. I don't mind. But so distinguishing between those two techniques is really important. Okay. Okay, the rest of the talk I want to talk about just drugs, which I think is it's a topic people usually want to hear about, right? So imatinib and beyond imatinib. So the first drug shown to be effective for GIST was imatinib. And we just saw how very effective it was in patient number 1. Unfortunately, it's not always that effective. There are some GISTs that for one reason or another just don't respond to imatinib. And there are cases where the tumor responds to imatinib, but the patient has a very bad reaction, and you really have to try something else. People are unique. Every person is different. You never know with any given drug, right? Some people have bad reactions to drugs that most people tolerate without any difficulty. It's an individual kind of thing. So one drug is never enough. You always need additional drugs for any condition. He speaks as though he were a doctor. But it's true, right? Okay. So more drugs were developed. In fact, the second and third line, I think they say in medicine, they tell doctors, you never want to be the first to try a new therapy or the last to abandon an old therapy. Doctors tend to be conservative, and that's not a bad thing. I mean, you don't want doctors to go around doing reckless, crazy experimentation. Doctors tend to be conservative in how they deal with things. And so the tendency is that you start with drug number 1. And if it isn't working, you go to drug number 2. And if that isn't working, go to drug number 3. I mean, that's how they tend to think, and it's probably correct that they do. And so there is a first and second and third line drug for GIST. I think many of you know that, or many of you are on these drugs, one of these drugs. And they really came along in the order that they were discovered and introduced. And so there's no magic as to what the order was. But imatinib worked so well and was the first drug, so it's the first line treatment. Like, why would you not use imatinib? It works for most patients. So no doctor is going to say, I think this patient, I'll start him on the third line drug just to see what happens. You can't do that. You always start at number 1. And so the first treatment is almost always going to be imatinib. And then, unfortunately, some patients don't respond, or they don't tolerate the drug well, or they respond for a while, and then they stop responding, and the tumor starts to grow again. That's when you switch to number 2. A couple of things to note about the first, second, and third line drugs. First of all, none of them was developed for GIST. They were all, as I already said about imatinib, it's true of all these drugs. They were all developed for other cancers and other oncogenes. It's maybe not surprising because GIST is rare, and in the early days, there weren't any pharmaceutical companies that were actually researching to develop drugs for GIST because it was too rare a disease. They were developing drugs for other cancers. But in each case, the similarity between their target and KIT was close enough that it turned out that the drugs worked okay for GIST. And so those are Sutent or sunitinib from Pfizer, regorafenib or Stivarga from Bayer. They are what people call Me Too drugs. They're Me Too drugs. I mean, imatinib was the first in the class, and the other drugs were developed as Me Too, imitation drugs, if you will, variations on imatinib. That's really what they are. They're chemical, pharmacological, they're variations on imatinib. That's not a bad thing. You need those additional possible drugs, right? So it's a good thing that they were developed. But 2 things. Number 1, they all act essentially by the same biochemical, pharmacological mechanism. They all do the same thing. A little bit differently, basically they all do the same thing. And number two, as I said, none of them was developed specifically for GIST. But they all work, better or worse. They all work to some extent. Okay. And what they do is they prevent the binding of a molecule called ATP, which we're not going to talk about here, but if you have the fun of taking a biochemistry class, you'll learn about ATP. So they all act by the same mechanism at the molecular level. And then, beginning in the around 2010 or so, some startup companies started up that were specifically targeting GIST, developing drugs specifically to target KIT. Companies like Blueprint in Cambridge, Massachusetts, were set up and started programs specifically to look for KIT. That's a startup kind of job because it's a big gamble, and it's a gamble on a small market. And some of those startups have been complete failures, like Theseus, that just crashed a year ago. It was just a complete failure, their drug. But luckily, some of them have worked out okay. And so after years of research and development, some of these drugs began to come on the market around 2020. And you may have heard of a couple of these. There's ripretinib, also called Quinlock, developed by a company called Deciphera, which was recently bought by a Japanese company called Ono. And there's a drug called avapritinib, which was developed by Blueprint and is still produced by them. I have to tell you, we had a lot of hope in the GIST community that these bespoke KIT-focused drugs were really going to be a revolutionary improvement over the existing drugs. And the fact is they weren't, okay? They're pretty good, but they weren't as good as people had hoped. And in particular, none of them has been good enough to displace imatinib as first line. So really, it remains first, second, third line drugs have not changed. That's slightly not entirely true. But basically, the first, second, third line drugs have not changed, and these drugs are still in fourth or fifth place. But it's a little more complicated than that, as the doctors will tell you. Okay. In particular, avapritinib turns out to be very good, but for one very specific genetic subtype of KIT called PDGFR8D842V, which is a rare subtype of GIST. But for that rare subtype, avapritinib is a drug to go to. Okay. This is a comment, it's a comment I heard years ago from a GIST doctor. And unfortunately, it's still a very true comment in many cases, I'm sad to say. He said, GIST patients do well until they don't do well, and then they don't do well. What he meant by that was, as long as the drugs are working, imatinib, for example, as long as it's working and it's halted the growth of the tumor, GIST patients do so well that most of their friends say, what do you mean you've got cancer? You look fine. You haven't lost your hair, you're still at work, whatever. They do well. Unfortunately, in many cases, after a certain number of years, the tumor develops resistance to the drug. It's almost inevitable, unfortunately, that after enough years go by, some clones of cells develop in the tumor that are no longer responding to the drug. And once that resistance begins, then you're on to second and third and fourth line drugs. And unfortunately, that can be a pretty difficult battle as you go to second, third, fourth line drugs. It gets harder and harder for biological reasons, right? And so the usual paradigm for the doctors, as I've already indicated, you start with imatinib until it isn't working anymore. And when the tumor starts to progress, then, well, maybe you can increase the dose of imatinib, and they try that, but you can only increase so far. You can't go too high or the toxicity is too bad. So if the dose escalation isn't working, then you switch to sunitinib until that stops working. and then you switch to regorafenib until that stops working. That's generally how it goes. Okay, this is another comment I heard from a GIST doctor years ago, and it stuck in my mind, and I still bear it in mind. This was Dr. Reichert, one of the experts in Germany, and he says, when I'm referred to GIST patient, this is what happens when imatinib fails, right? When imatinib is no longer working. He said, when I'm referred to GIST patient because he or she has progressed while on imatinib, I first stop and ask 3 questions. Was it really a GIST? Because there are still misdiagnoses, even though there shouldn't be anymore, but there are. Was it really a progression? Because diagnosing the progression has happened is not so easy. It depends on careful interpretation of the radiology and the medical results. And doctors can, unfortunately, we still see cases quite often where doctors dismiss a drug as having failed too early, when it really was still working, but they just got anxious and switched too quickly. We think that still happens. And thirdly, was the patient really taking the drug? Because there are cases, for whatever reasons, of non-compliance with the drug. So these are caveats that you want to bear in mind when they say the disease is progressing. Are you really sure? Okay, so why does the drug stop working? Typically, it's because in addition to the initial mutation that turned on the KIT activity and started the tumor, now additional mutations have cropped up which are conferring resistance to the drug, okay, typically to imatinib. It's extremely complicated because it's a long gene and there are many different parts of the gene that can undergo mutations. It's really, really complicated. But to simplify it, this is, so the long bar from top to bottom represents the KIT gene with primary and secondary mutations. And then we have this simplified traffic light pattern of red, yellow, or green lights, which indicate for each of these drugs, imatinib, sunitinib, regorafenib, ripretinib, the first, second, third, fourth line drugs, are the tumors that have these particular additional mutations, are they sensitive to that drug or resistant to it? And you'll notice that the first column for imatinib, every traffic light is red. That's by definition, because these are imatinib-resistant mutations. So of course they're all red. They're red because secondary mutations have occurred which conferred imatinib resistance. So imatinib is no longer working. What really is really crucial is, what about the other drugs? Do we have any green lights? And you'll see for sunitinib there are some, but not all the mutations are sensitive. You want a green light, which means sensitivity. But there are still red lights even with sunitinib. The hope is that as you read across that one of those rows, that you will eventually see a green light, a drug that will still work against that secondary mutation. But this is very oversimplified. It's not really all red or green, right? It's a lot more subtle than that. But this is a rough guide. Okay, yeah. So they're all resistant to imatinib, and we're hoping. Okay, so I'm coming to the conclusion, which is good because I'm running out of time. But what we would like to see is more green lights. More drugs, more green lights, more effective drugs. In fact, ultimately, you'd like to see a drug that was even better than imatinib in first line. It may never happen because imatinib is so good in first line that to challenge it, a drug company would have to fund a clinical trial of taking newly diagnosed patients and treating them either with imatinib or with new drug X. And they're never going to do that because we know imatinib works. So it's just not ethical not to give patients imatinib, basically. So you're probably never going to see that trial. for the foreseeable future. It's always going to start with imatinib. The question is, can we challenge in second or third line? And that's what people are really trying to do now. And in particular, informed by knowing what the secondary mutation is, if we know it. So how do you know what the secondary mutation is? That's not an easy question. So there are techniques you may hear of at the meeting, like ctDNA, circulating tumor DNA, that may help us to figure out what the secondary mutation is. Not a trivial question at all. But maybe if we can be guided by knowing what the secondary mutation is, then maybe if and when imatinib fails, maybe we can jump to the best drug rather than a drug that is not so good. And that's what people are trying right now in clinical trials. Can we optimize at least the second-line treatment? And beyond that, what we want are better drugs. And why do we need better drugs? For all these reasons. As I've said, some GISTs don't respond to imatinib even from the outset. They're imatinib resistant for whatever reason right at the beginning. Tolerance of the drugs is highly variable. There are patients who take 800 or even 1,000 milligrams of imatinib every day and do okay. And there are other patients who can't tolerate 100 milligrams of imatinib. It's highly variable, right? And why? Well, I don't really know. It's just everybody, each person is unique. People have different properties in every way: weight, diet, ethnic origin, genetics, whatever, digestive issues, whatever, all kinds of different reasons. Thirdly, as I've already said, TKIs. I mean, what does a TKI do? It really stops the growth signal that is being transmitted by the KIT protein. It stops the cell dividing. But that's very different from traditional chemotherapies that actually kill cells. You know, the traditional cytotoxic chemotherapy, hair falling out, side effects of cancer treatment, those are usually toxic drugs that actually kill cells. Maybe not very effectively, but they really kill the cells. These drugs, which we call TKIs, tyrosine kinase inhibitor drugs, all the ones I've been talking about, they don't really kill the cells. They just put them to sleep, and maybe not completely to sleep. Okay? So that's why these cells, even with drug treatment, there tends to be a little bit of cell division and a little bit of development of new mutations in an ongoing basis, because you can't stop them completely from dividing. So ultimately, really, we've yet to find a case, I would say, where ultimately you don't have to face this challenge of TKI resistance. It limits the usefulness of every new drug. So we need new drugs. And as you'll see at this meeting, new drugs are still being developed, which we hope will help a lot. Okay? Okay. I think this is the last slide, almost the last slide. Last couple of slides, and the new drugs. You'll hear about these at the meeting. I'm not going to say anything about them in detail, but these are new KIT inhibitors that are coming out. So there's this drug developed at AstraZeneca now from a company in China called NewBay, NB003. There's IDRX. I think are they here at the meeting? So it's a drug from IDRX. And there's this drug developed by a company called Plexicon, now marketed by Cogent. And that's the PEAK trial that I think, are they here? They're here. Cogent is here. They have a table here, so you'll hear about that drug too. And you'll hear about this trial, you'll hear about from the Cogent people. And I think, okay, I think I'll really leave it at that point because we're running out of time. Okay, thanks. That's a really important question. So, I mean, what we're asking about here is, it's another important topic I just didn't have time to talk about, right? So, if the tumor is localized, surgery was successful, the margins were clear, the tumor didn't rupture, some doctor, the fundamental attitude of the surgeons is, bye, have a good life, nothing to worry about, I'll never see you again, right? That's true for most tumors, right? Oh, you're cured. I took it out. But then some doctors, notably Dr. José in Finland, said, maybe that's not good enough. Maybe we should try, even after successful removal of a localized tumor, let's see if giving the patient imatinib in a preventative way might actually delay progression or recurrence. Because it was known that even when you do take out a GIST, there's a certain chance that it will recur or progress later, because you can't get every cell out, right? And so it turned out that did work pretty well, and we call that adjuvant imatinib therapy. And it is a weird thing to do, because it means you're treating patients who are well. They have no evidence of disease, but you're still giving them the drug. And then the question is, how long do you go on for? And that's a really difficult question, and we still don't really know. The standard now is 3 years, right? Once it's progressed or recurred, then it's no longer adjuvant treatment. I mean, then it is in the same category as a metastatic tumor. Well, the question is not whether the mutation is there, but whether the cells are still there, right? I mean, if the tumor has been successfully resected and taken out, then the fundamental attitude of the surgeon is, it's done, you're cured, there's no cancer cells left. But of course, we know that's usually not really true, but well, maybe so. I mean, a lot of times it is true. I mean, something like 50% of GISTs are localized when they're diagnosed and they're successfully resected. I know lots of patients in Canada anyway who were treated surgically, localized, successful surgery, and literally, like I just said, the surgeon says, you're cured, bye. And they say, well, when's my follow-up appointment? And they say, why do you want to follow up? You're cured, bye. We'll see you again. I mean, I don't think that's the right thing to say, but there are still lots of surgeons who say that you're cured, bye. No, don't even think about it again. And I mean, sometimes, and for a lot of patients, that's true. It never comes back from a localized case. That's why, like I said at the beginning, localized versus metastatic, those are two different paths, very different roads, right? Totally different. So the question is, when it is localized, can we predict the likelihood that it will recur? And that depends on size, something called mitotic rate, whether the tumor ruptured, and some various other factors. So that's a whole really important question that I just didn't talk about at all. But I mean, the only reason you do that, that's called risk analysis, right? What is the risk of recurrence? But the only reason to do that is to decide whether and for how long to do adjuvant treatment. I mean, there's no other reason to do it, or maybe also to guide how frequently you want to bring people in for CAT scans and so on. But I mean, that risk analysis is not going to stop what happens. It's just going to give you a better estimate of the likelihood that it will happen, right? It doesn't change the treatment, really. It may change the monitoring. I mean, like for really high risk, like if the tumor had ruptured or ruptures during surgery, then that's automatically high risk because those cells have been just spread out, right? And so then clearly it makes sense to do frequent CAT scans or re-examinations to see what's going on, right?